Doing Edexcel AES level Biology B, and we are, in under 5 minutes, plan to cover protein structures. Okay, let's start. So, protein structures. The secondary, tertiary, and quaternary structure all depend on the sequence of amino acids, so-called the primary structure. When a polypeptide or chain of polypeptide fold, a functional protein is produced. Okay. Hydrogen bonding. Amino and carboxylic acid group in the amino acid chain carry small amounts of charge. This is delta negative on the COO of the carboxylic group and delta positive on the NH of the amino group. Right, let me just explain that. These charges result in hydrogen bonds forming between these parts of the chain, which stabilize the structure. Okay, so all you need to know is NOF. NOF is nitrogen, oxygen, and fluorine. Whenever they are bonded to a hydrogen atom, they have the ability to form hydrogen bonds, and this is because electronegativity is a concept in covalent bonding, so that's between two nonmetal atoms, where when you're bonded to one of these three atoms, they have a stronger ability to pull the electrons slightly closer to them. Now, we know electrons are negative, and so the electrons are slightly closer to them. They will have a slightly negative charge known as delta negative. Delta just means slightly, and so remember, they're not going to be in the middle. You're going to have an electron here and an electron here. Now, it's slightly to the nitrogen, and because electrons are negative, you're going to have a slightly negative charge called delta negative. This would in turn mean that the hydrogen has become slightly positive, hence called delta positive. Now, if we go to now this bit, we have carbon and an oxygen that's bonded together. Now, what did I say? When it's an oxygen atom, it can an oxygen atom, that's going to be slightly delta negative and slightly positive, because whenever you have one of these three atoms, they can pull the electrons from another atom, but mostly you see it occurring with a hydrogen, because now you can form a hydrogen bond between these two, because that's delta negative, that's delta positive. Same thing has happened here. And this thin dotted line is what we call a hydrogen bond, and that helps to stabilize structures. Okay, it's nitrogen, oxygen, and fluorine. Whenever they're bonded to another atom, particularly hydrogen, there has to be hydrogen involved, and you will have the presence of hydrogen bonding. Right. Secondary structure. The secondary structure is where polypeptide chains are held together by many hydrogen bonds, and this results in two types of shapes occurring. You can either get an alpha helix shape or a beta pleated sheet. You don't need to know what these structures in detail are, as written here. All you need to know is it turns into an alpha helix, which is a helix shape, or beta pleated sheets, which is basically, it looks like this, like sheets. That's all you need to know for the secondary structure, and you have more hydrogen bonding. Now, we move on to the tertiary structure. In the next level of organization, the alpha helix or the beta pleated sheet will result in further folding, and that gives rise to this tertiary structure. Now, once we have a tertiary structure forming, we will have different types of bond coming in. We have ionic bonds, and that's between R groups. That's just the R group is what differentiates amino acids from each other, and it can be any type of group here. And so we have ionic bonding. We also have disulfide bonds, also known as disulfide bridges, and that's when you have bonds between SH groups. That's, for example, like between cysteines. So whenever you see S and S, that just basically means disulfide bridges or disulfide bonds. Now, so a protein folded into a tertiary structure held together by hydrogen bonds, ionic bonds, and disulfide bonds, and that's all between the R groups. Now, the quaternary structure. In some proteins, a small number of separate, often different, folded chains join together to form a quaternary structure. Each folded chain is joined to the others by the same range of bonds as for the tertiary structure bonding. What that means is that in the quaternary structure, all you have is two or more tertiary structures that are joining together, and they join together by hydrogen bonding. Right. Explain how the sequence of amino acid determines the shape of a protein with reference to insulin. Okay, you will never know the structure for insulin because you don't cover it, but you do know the sequence of amino acids for the shape. So amino acids have different R groups. This determines which bonds can form between them in the chain. For example, we can have disulfide bonds, ionic bonds, and hydrogen bonds. Now, compare and contrast the ways in which secondary, tertiary, and quaternary structures are held together in proteins. Okay, so they all have hydrogen bonding. However, the secondary and the quaternary structure only has hydrogen bonding forming, whereas in the tertiary structure, you will have the formation of hydrogen bonds, ionic bonds, and disulfide bridges. So that's the main difference between them all. And that's it.